Homologous recombination requires extensive regions of sequence similarity. Whether the end product is DNA repair or the exchange of nucleotide sequences during meiosis, the hallmark of homologous recombination is that it takes place only between DNA duplexes that contain extensive regions of sequence similarity, homology. A pair of DNA molecules can assess this homology by sampling each other's nucleotide sequences when a single strand from one DNA duplex engages in an extensive bout of base pairing with the complementary strand from the other duplex. The match need not be perfect for homologous recombination to succeed, but it must be very close. Homologous recombination can flawlessly repair DNA double-strand breaks. Homologous recombination is often initiated when a double-strand break occurs shortly after a stretch of DNA has been replicated. At that time, the duplicated helices are still in close proximity to one another. Figure 629a. To begin the repair, a nuclease generates single-stranded ends at the break by chewing back one of the complementary DNA strands. Figure 629b. With the help of specialized enzymes, one of these single strands then invades the homologous DNA duplex by forming base pairs with its complementary strand. If this sampling results in extensive base pairing, a branch point is created where the two DNA strands, one from each duplex, cross. Figure 629c. At this point, the invading strand is elongated by a repair DNA polymerase, using the complementary strand as a template. Figure 629d. The branch point then migrates as the base pairs holding together the duplexes break and new ones form. Figure 629e. Repair is completed by additional DNA synthesis, followed by DNA ligation. Figure 629f. The net result is two intact DNA helices, where the genetic information from one was used as a template to repair the other. Homologous recombination can also be used to repair many other types of DNA damage, making it perhaps the most versatile DNA repair mechanism available to the cell. All that is needed is an intact homologous chromosome to use as a partner, and such paired identical chromosomes are produced transiently each time a chromosome is replicated. The all-purpose nature of recombinational repair probably explains why this mechanism and the proteins that carry it out have been conserved in virtually all cells on Earth. Homologous recombination exchanges genetic information during meiosis. Sexually reproducing organisms depend on the process of meiosis to generate germ cells, sperm and eggs in the case of mammals. Homologous recombination is essential for this process to proceed properly, as we discuss in more detail in Chapter 19. A particularly important consequence of homologous recombination during meiosis is the formation of chromosomal crossovers. Here, two homologous chromosomes, one from the father, one from the mother, come together and undergo a genetic exchange. Figure 630. The site of exchange can be anywhere in the homologous nucleotide sequences of the two participating DNA molecules. The cleavage and rejoining events that mediate the exchange occur so precisely that not a single nucleotide is lost or gained. Because the paternal and maternal chromosomes typically differ slightly in their DNA sequences, their crossing over generates new combinations of DNA sequences in each chromosome. The benefit of such gene mixing for the progeny organisms is apparently so great that the reassortment of genes by homologous recombination is not confined to sexually reproducing organisms. It is also widespread in asexually reproducing organisms. For example, when a bacterium acquires a homologous chromosome from another bacterial cell through horizontal gene transfer, as we will see in Chapter 9. Homologous recombination during meiosis begins with a bold stroke. A specialized enzyme deliberately slices through both strands of the recombining chromosomes, creating a double-strand break. At this point, some of the same proteins that function in recombinational double-strand break repair converge on the damage. However, these recombination proteins are now directed by meiosis-specific proteins to perform their tasks differently, producing, via one or more crossovers, two molecules with novel DNA sequences. Figure 631. This outcome is possible because, in meiosis, recombination occurs preferentially between maternal and paternal chromosomes rather than between newly replicated identical DNA strands, as happens when homologous recombination mediates double-strand break repair. Crossing over during meiosis ensures that each of our chromosomes contains a combination of DNA sequences from our two parents. As we discuss in detail in Chapter 19, this type of chromosomal rearrangement generates large amounts of genetic diversity in the offspring of sexually reproducing organisms, and it has thereby contributed greatly to the stunning variety of life forms present on the planet.